All right, let's dive into something pretty small today.
Sounds fun.
It's biomolecule labeling, and I've got a stack of articles here that listeners sent in.
Yeah.
And honestly, it looked pretty complex, so I'm really glad you're here.
It is a complex field, but it's super fascinating, too. If you think about it, it's like attaching a microscopic tracking device to a single molecule.
Okay, so we're basically talking about like spying on molecules?
Basically, yeah.
That's pretty wild. But why go through all that trouble? What's the benefit of labeling these tiny things?
Well, it lets us see where they go, how they interact with other molecules, even measure how much of a specific molecule is present.
Hmm.
That's really important if you want to understand how cells work, for example,
Yeah.
or develop new drugs or diagnose diseases.
So this isn't just some abstract science. It actually affects our lives.
Absolutely. Almost every major medical advance you can think of probably relied on biomolecule labeling at some point.
Wow. Okay, I had no idea it was so widespread. So what do they actually use for these tracking devices? I mean, what are we talking about here?
Well, there are a bunch of different options, each with their own advantages and disadvantages. Some common ones are fluorescent dyes.
Okay, yeah.
They light up under a microscope, and biotin, which you can think of like a molecular hook.
Oh, that's
It
interesting.
can grab onto other molecules.
Clever.
Yeah. And then there are radioactive isotopes that give off a detectable signal.
Mm-hmm.
And then even enzymes, which can cause a measurable change when they react with specific molecules.
So it's a whole range of techniques then.
Right. And the choice depends on a lot of things,
Like what?
like the size of the molecule or where it's located in the cell.
Okay.
And of course, what information you're trying to get.
Makes sense. One of the articles you sent in talks about labeling proteins.
Right.
Those are the big, complex molecules that do all kinds of work in the cell.
Exactly. They're crucial for pretty much every biological process.
And I imagine they're hard to label.
They can be, yeah. You have to be careful not to mess up their function when you attach a label.
Right. Because I guess if you disrupt the protein, then your results won't be accurate.
Yeah, you've got it.
So how do they do it?
There are two main approaches: site-specific labeling and random labeling.
Hmm.
Site-specific labeling is like putting a GPS tracker on a specific spot.
Hmm.
You're using enzymes or reactions that target very specific amino acids within the protein.
Oh, so it's very precise.
Yes, yeah.
Yeah.
You know exactly where that label's going to end up.
Okay. What about random labeling?
It's a bit more like scattering breadcrumbs. You're using reactions that can attach the label to any available functional group on the protein.
So it's faster, but maybe less accurate?
It can be. Yeah, you run the risk of the label interfering with the protein's function.
So it's a trade-off.
Exactly. Researchers have to weigh the pros and cons of each method.
So we've talked about the big guys, the proteins. What about the small molecules? How do you label something that tiny?
That's a whole other challenge. Small molecules are, well, small, and they're often very sensitive to changes in their structure.
I see.
So you have to be really careful not to disrupt their function when you attach a label.
So how do they do it? What are the tricks of the trade?
Well, some of the common strategies are direct attack and chelation.
Direct attack.
It sounds dramatic, right?
It does.
But it's not that complicated. Basically, it means targeting specific functional groups that are already part of the molecule.
So they're using what's already there.
Yeah, things like amines, carboxyls, and thiols. They act like little handles that scientists can use to attach labels.
I see. So what about this chelation thing?
Chelation is more like building a tiny cage around the small molecule
Okay.
to hold the label in place.
Interesting. So containment rather than direct attachment.
Exactly. And this is particularly useful when you're using radioactive labels because they can be a bit tricky to handle.
Yeah, I can imagine.
This cage helps to keep them contained and prevents them from reacting with other molecules.
So we're talking about some pretty sophisticated molecular engineering here.
Oh, absolutely. The techniques for labeling biomolecules have become incredibly advanced, and they're constantly evolving.
Yeah, one of the articles mentions click chemistry.
Right?
It sounds so futuristic.
It's a really cool technique. It's all about using very specific reactions that work even inside the complex environment of a living cell.
Okay, but how do they get these tiny molecules to connect with each other in that chaotic environment? That just seems mind-blowing to me.
It is pretty amazing, and we'll dig into exactly how it works in the next part of our deep dive.
Sounds good. I'm definitely intrigued to learn more about that.
Click chemistry, it's really remarkable. It's about using very specific reactions that work efficiently, even inside a living cell.
Okay, so we're talking about these tiny molecules floating around in a cell, and somehow they manage to find each other and click together.
Right. And it works because the reactions used in click chemistry are bioorthogonal.
What was that again?
Bioorthogonal, it means they're highly specific. These reactions basically act like a molecular handshake that only occurs between two designated chemical groups.
So like a secret code that only these two molecules know.
Exactly. That ensures they connect with each other and nothing else.
That's so cool. So it doesn't interfere with any of the other stuff happening inside the cell.
Right. It's like they have their own little communication channel.
That's incredible. So we're talking about tracking molecules in real time as they're doing their thing inside the cell?
Yeah, pretty much. It opens up a whole world of possibilities for research.
Like what?
Well, you could study how proteins interact with each other,
Okay.
or track the movement of drug molecules as they target specific cells. You could even see how viruses hijack cellular machinery to replicate themselves.
That's amazing. It's like having a front row seat to the microscopic world. Speaking of cool techniques, what about halo tag technology?
Ah, halo tag. That's another great one. It's super versatile.
How so?
Well, remember how we talked about the challenges of labeling different types of molecules?
Yeah.
Halo tag offers a universal solution.
Really?
Yeah, it all starts with genetically engineering a special protein tag called the halo tag onto your protein of interest. This tag acts like a docking station
A docking station for what?
for a molecule called a ligand. The ligand binds very specifically to the halo tag.
Huh?
And here's the clever part. You can attach pretty much any label you want to this ligand.
So it's like a modular system?
Exactly. You've got the halo tag as the base, and then you can just swap out different ligands with different labels depending on what you need.
Oh, I see. So if you need to track a protein's movement, you use a ligand with a fluorescent dye.
Right.
But if you want to purify a protein, you use a ligand with a magnetic bead or something.
Yeah, it's a very flexible system.
This is amazing. It sounds almost too good to be true. Are there any downsides to these techniques?
Well, no technique is perfect. Of course, there are limitations.
Like what?
Well, with click chemistry, you often have to introduce unnatural chemical groups into the molecules you're studying. And sometimes, though not always, that can affect the molecule's normal function.
So it's like a trade-off between the precision of the technique and the potential for disruption.
Yeah, you have to be aware of that. And then with halo tag, the main limitation is that you have to genetically modify your protein to include the halo tag,
Right.
which isn't always possible or desirable.
Especially when working with, like, complex organisms or something.
Exactly.
It sounds like scientists have to walk a fine line.
They do. It's always a balancing act. But that's what makes this field so exciting.
It is, yeah. There's always something new to learn and new challenges to tackle.
Mm.
Okay, so we've talked a lot about these advanced techniques, but I keep coming back to that basic question. Why should we care? How does this actually affect us?
It's a great question.
Ah,
And the answer is that biomolecule labeling has real-world applications that are changing the world.
Yeah.
Think about cancer research.
okay.
Biomolecule labeling is helping us develop new cancer therapies, track cancer cells, and even design targeted drug delivery systems.
So more effective treatments with fewer side effects.
Exactly. It's about improving the lives of people with cancer.
That's incredible. And what about other diseases?
Well, it's also revolutionizing our understanding of neurological diseases like Alzheimer's and Parkinson's.
Wow.
We're using it to unravel the complexities of the brain and develop new therapies for these devastating conditions.
That's amazing. So by looking at these tiny molecules, we can gain insights into the brain.
We can. It's incredible. And it's not just treatment. Biomolecule labeling is also transforming how we diagnose diseases.
Oh, right. You mentioned that before.
Yeah, it's leading to more sensitive and accurate diagnostic tests, which means earlier detection and better outcomes for patients.
So we're talking about potentially saving lives.
Absolutely. It's amazing to think that something so small can have such a huge impact.
It really is. This deep dive has been so eye-opening. I'm in awe of what scientists are doing in this field.
It is a remarkable field. It's a testament to human ingenuity.
I totally agree. I'm feeling a bit overwhelmed by the complexity of it all, though.
That's understandable. It's a lot to take in. But that's the beauty of science. There's always more to learn.
That's true. So where do we go from here?
Well, we've talked about the what and the why of biomolecule labeling,
Right.
but there's also the how.
Ah, yes. Click chemistry, halo tag, all that good stuff.
Exactly. Let's delve into the nuts and bolts of those techniques and see how they're being used in real-world research.
Okay, let's do it. Okay, so let's get into the lab.
time for some experiments.
Well, metaphorically, of course, I'm ready to learn how this stuff actually works.
Yeah.
How do they attach these labels to the molecules?
It's a really neat blend of chemistry and biology. Remember we talked about site-specific and random labeling?
Yeah.
Each approach uses different tools and techniques.
Okay, and what about click chemistry and halo tag?
We'll definitely get to those.
Those are the ones I'm most curious about.
I bet. Let's start with site-specific labeling. This is where precision is key. It's like you want to place a beacon on a very specific spot.
I see.
And one way to do this is to use enzymes.
Right, those tiny molecular machines.
Exactly. They can catalyze very specific reactions.
Hmm.
And they're enzymes that can attach labels to very specific amino acids within a protein.
Wow, so you can really control where the label goes.
Yeah, you can. It's super important for experiments where you need to know the precise location of a molecule.
Like tracking its movement within a cell or something.
Exactly. And there are other site-specific methods, too.
Like what?
Well, scientists can also design chemical reactions that target unique functional groups on the molecule.
So they're exploiting the specific chemistry of their target.
Yep, they're taking advantage of what's already there to make sure the label ends up in the right place.
That's really clever. So what about random labeling?
Random labeling is more about speed. Sometimes you need to label a lot of molecules quickly, and you don't necessarily need to know exactly where the label goes.
It's like casting a wider net.
Exactly.
Yeah.
And in some experiments, the exact location of the label doesn't really matter.
Okay, that makes sense. So when would you use click chemistry then?
Click chemistry is great when you need a really specific and efficient reaction. One of the most popular ones is called the copper-catalyzed azide alkene cycloaddition.
Wow, that's a mouthful.
It is, haha. We can just call it CoAC for short or just stick with click chemistry.
Yeah, click chemistry is easier to remember. So how does it work?
You have one molecule with a special chemical group called an azide and another with a group called an alkene. And these two groups are like puzzle pieces. They're designed to fit perfectly together.
Okay.
When you add a copper catalyst, it brings them together and they snap into place, forming a stable link between the two molecules.
So the copper is like the matchmaker.
Yeah, that's a good way to put it.
And this reaction is super efficient and specific.
It is, yeah. It works really well even inside living cells, and it doesn't mess with other biological processes.
That's amazing. Okay, so how about halo tag? How does that one work?
Halo tag is all about customization. You start by genetically engineering a special protein tag onto the protein you're interested in.
This is the halo tag, right?
Yep. And this tag acts like a universal docking station for a molecule called a ligand. It binds to the halo tag very specifically.
Okay.
And the cool thing is you can attach any kind of label you want to this ligand.
Oh, so it's like a build-your-own labeling system.
Exactly. You need a fluorescent label. No problem. You want to use a radioactive isotope. Go for it. It's incredibly flexible.
Wow, that's really impressive. But I imagine there are some challenges too, right?
Of course.
Yeah.
There are always challenges in science.
Oh,
One of the biggest is making sure that the label doesn't interfere with the molecule's normal function.
right, because then your experiment is kind of pointless.
Exactly. So scientists have to choose labels that are small and inert, meaning they won't react with other things in the cell.
And I guess the labeling process itself has to be carefully controlled too.
Oh, definitely. You don't want to damage the molecule you're trying to study.
So it's balancing act, getting the label attached without messing things up.
It is.
Huh?
And another challenge is making sure that the label is specific to your target molecule.
Right, especially in complex environments like a cell.
Exactly. You don't want your label attaching to random things and giving you false results.
So how do they make sure that doesn't happen?
Well, they're constantly developing new techniques to improve the specificity of labeling. Sometimes they use antibodies, which are really good at recognizing specific targets, or they design labels that only become active when they're bound to the target molecule.
Oh, that's clever. So it's like a constant race to improve these techniques.
It is. It's a really dynamic field,
Yeah.
and there are always new breakthroughs happening.
Well, this has been an incredible journey. We've covered so much ground, from the basic concept of biomolecule labeling to these cutting-edge techniques that are revolutionizing research.
It's been a lot of fun, hasn't it?
It has. I've learned so much. I'm feeling really inspired by the ingenuity and dedication of these scientists.
Yeah, they're doing some amazing work.
And who knows what the future holds for this field. It seems like there's so much potential for new discoveries and applications.
Absolutely. It's an exciting time to be following this research.
And for our listeners who want to dive deeper into this topic, we've included some links in the show notes.
Yeah, check those out.
So keep those brains buzzing and never stop learning.
Stay curious.
Thanks for joining us on this deep dive, everyone. Until next time.